Good morning, everyone. Our topic for today is all about the process of mitosis and meiosis. Mitosis is the process where a single cell divided into two identical daughter cells. And we have a meiosis is divided into five phases. Here is interphase, prophase, metaphase, anaphase, and telophase. Number one is interphase. What is interphase? Interphase is the parts of the cell. This is when the cell grows and copies its DNA before moving into meiosis. Interphase during meiosis, chromosomes with a line separate and move into daughter cell. And we have a prophase. It's the first phase of mitosis, the process that separates the duplicate genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prophase, the complex of DNA and protein contained in the nucleus known as chromatide condenses. Next is metaphase. Metaphase is the third phase of mitosis, the process that separates duplicate genetic material carried in the nucleus of a parent cell into two identical daughter cells. Next is anaphase. Anaphase is the fourth phase of mitosis, the process that separates the duplicate genetic material carried in the nucleus of a parent cell into identical cells. The separated chromosomes are then followed by the spindle to opposite poles of the cell. Next, we have a telophase. Telophase is the fifth and final phase of mitosis, the process that separates the duplicate genetic material carried in the nucleus of a parent cell into two identical daughter cells. During telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. And meiosis. Meiosis is a special type of cell divisions of germ cells in sexually reproducing organisms used to produce the gametes such as sperm or egg cell. There are two stages or phases of meiosis, meiosis 1 and meiosis 2. The first phase of meiosis interphase, known as G1, cells grow and perform many of their required cellular functions. Next is prophase 1. Prophase 1 is essentially the crossing over the recombination of genetic material between non-sister chromatids that this results in the genetically identically haploid daughter chromatid cells. Next is the first metaphase of meiosis 1, encompasses the alignment of paired chromosomes along the center metaphase plates of the cell, ensuring the complete copies of chromosomes are present in the resulting two daughter cells of meiosis 1. Next is we have in anaphase 1. Anaphase 1, the homologies are pulled apart and move apart to opposite ends of the cell. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart. Next is we have a telophase 1. Telophase 1 is the phase when the chromosomes have finished among the opposite end of the cell. This will then be followed by cytokinesis producing two daughter cells. Next is interphase. We have an interphase 2 where to add further DNA replication takes place. During meiosis 2, chromosomes align at the center of the cell in metaphase 2 exactly the way they do in mitotic metaphase. In anaphase 2, the sister chromosomes are separated again, the same function as in mitotic anaphase. Next in meiosis 2 is prophase 2. Chromosomes condense and nuclear envelope break down if needed. The centrosomes move apart, the spindle forms between them, and the spindle microtubules begin to capture chromosomes. Next is metaphase 2, and meiosis 1. The chromosomes line up individually along the metaphase plate. Next is anaphase 2. Anaphase 2, the sister chromatids separate and are pulled toward opposite pole of the cell. And we have a telophase 2. The fourth step on meiosis 2, the chromosomes reach opposite poles. Cytokinesis occurs. The two cells produced by meiosis 1 divided to form fourth haploid daughter cell, a nuclear envelope, while in the diagram of at right form. Next, and this is for today. Thank you.